Friends, welcome to Biology Made Easy. Today in this video we'll discuss about the special type of chromosomes, or you can call them as giant chromosomes. Giant chromosomes, or you can tell them as special chromosomes. So why they name giant chromosomes? Because they are quite big in size than the standard chromosomes. The chromosomes which are present in the normal organisms, in the normal cells, they are the standard chromosomes. But these chromosomes are quite big in size. That is why they are known as giant chromosomes. So the giant chromosomes, mainly we will discuss here two types of chromosome. One is a polytene chromosome, and the other is the second one is that is a Lamb-Brush chromosome. These two will discuss here. Now the polytene chromosome was discovered by Balbiani in 1891. Usually these polytene chromosomes, they are present in the salivary gland, of Drosophila, then in nerve cells, then the larval tissues of dipterous flies, they are also present in the intestinal cells of larva of mosquito. So they are usually present. Now these polytene chromosomes, they are the special chromosomes present in the interphase stage. And usually they are in a somatic pairings. They are always in a state of somatic pairing. And their length is 100 times larger than normal chromosomes. Usually, since they are present in the somatic pairing in the salivary gland of Drosophila, there are 4 polytene chromosomes. And they are attached to a common center known as chromocenter. That means suppose this is one point, and to this the 4 polytene chromosomes are attached. That means this point is known as chromocenter. Now the question is, why they are known as polytene chromosomes? So poly means many, teen means the chromatin. So when it is polytene chromosomes, that means usually in each chromosome there are 2 chromatids. Now suppose there will be a duplication of these chromatids. This will become, the 2 will become, or the 1 has become 2 during a space, but suppose they will not be separated. And now again both of them will be duplicated or replicated. So it will be 4. And without separation again they will be duplicated, then 8, 16, 32 like that. That means there is a duplication of the chromatids without separation. So this gives, that means they are formed due to endomitosis. So endo means within. So within the nucleus. That means within the nucleus there is mitosis. That is without any cytokinesis, and the nuclear membrane is not dissolved. So this is known as endomitosis, and it gives rise to some thousands of chromatids. And there are more length. It is because sometimes they are unfolded. to give rise to some unfolding structures which are known as chromosome puffs or Balbiani rings. Suppose this is one chromosome, and at a certain places it will be unfolded like this. That means you will there are certain dark bands. At these places the DNA is more folded. So at these places the DNA is more folded, but at certain places there is unfolding, and this is known as, according to name of Balbiani, this is known as Balbiani ring, or this is also known as chromosome puffs. This is known as chromosome puff. So at these chromosome puffs, the DNA is unfolded. That means this is the site of the replication. The site of replication, or also you can the transcription of RNA. Transcription of RNA takes place at these places, because at these places it is completely unfolded. So this is about this polytene chromosomes, and they are present only in some special organisms and particularly special nuclei, as we have given at these examples, and because of their size they are known as polytene chromosomes. And this way chromosome puffs also can be artificially produced from these polytene chromosomes by inducing the or giving the hormone ecdysone. Ecdysone. If ecdysone hormone is given to this polytene chromosome, then chromosome puffs can form, and here transcription RNA takes place. So this is about the polytene chromosomes. Now let us discuss the second one, that is the Lambrusche chromosomes. The Lambrusche chromosomes were discovered by Ruckert in 1892 from the Sark, and later on it was found that this is found in the diplotin stage, stage of primary oocyte of many animal species. It is also found in the giant nucleus, of Acetabularia. And this Lambrusche chromosome is much more bigger than that of the polytene chromosomes. That means bigger than polytene chromosomes. Bigger than polytene chromosomes. But diameter is less. Because here there are more number of chromatids, but yet less number of chromatids. So the diameter is less, but the length is much more than that of the polytene chromosomes. Now what is this? Why it is known as Lambrusche chromosome? Now you see, this is found in the diplotin stage. Diplotin is one of the substages of prophase 1. And before that, the crossing over has already taken place. That means the two chromosomes, they are in a pairing stage. So two chromosomes are in the pairing stage, and they are present like this. That means these are the different types of loops. So throughout this structure is present like this throughout. Will not draw the entire one. For example, throughout it will be like this. Now if you will expand a particular portion of this, then how it will look like? Like this. If you will expand a particular portion, it will look like this. That means suppose a particular portion will be expanded, it will look like this. So this is the loop where the DNA is completely unfolded, while this is the axis, and here the chromosomes are present in the folded manner. Now, why it is known as a Lambrusch chromosome? Because it resembles to the brush used in chimneys. That is why it is known as a Lambrusch chromosome. And in the loop portion, since the DNA is completely unfolded, at this portion there is a synthesis of mRNA. And why this mRNA is synthesized? Because it is in diploid stage. And in a diploid stage, usually there is no metabolic activity. But after this diploid stage, then there will be diakinesis, then there will be other stages, metaphase 1, anaphase 1, and it will be completed. And if the egg will be fertilized, it will give rise to the development of zygote and then embryo. During development of embryo, it requires much more proteins for quick growth. But how in a short time such a large number of proteins will be formed? That is why in those cases, as the example found in oocyte of many animal species, usually now this mRNA which is present, this mRNA is stored to be used in future in case of when the fertilization will take place for formation of embryo. But one thing is, this mRNA cannot stay for a long time. So, how this mRNA remains for future use? Usually this mRNA is covered by a protein. Now it is covered by a protein. And now this structure is known as informosome. So informosome means the mRNA surrounded by a protein coat. Now in this informosome will be stored to be used in future during the development of embryo. So that is the importance of this transcription by the loops in the diploid stage, where they will be utilized for the synthesis of more amount of proteins during the development of embryo. So these are the two special type of chromosomes. One is polytene chromosome, the other is Lambrusch chromosomes. Now you have some idea regarding the two chromosomes, and whatever question will be asked on these two aspects, you will be able to answer also. So thank you very much. Wish you a very bright future. God bless you.